All right, so I've prepared some slides to review Unit 6. This is chapters 14, 15, and 16. And in many ways, you might think of this unit as really culmination of everything we talked about all semester. So in Unit 1, 2, and 3, and I guess 4 as well, we talked about different characteristics of microorganisms. Actually, 1, 2, and 3, we talked about characteristics of microorganisms. Chapter 4 and 5, or maybe just 5, we talked about the diversity of microorganisms. And in this unit, we really have to use all of that information to be able to do what we're doing in Unit 6, because Unit 6 is how our body recognizes and fights off pathogens or even any microorganism. So we really have to fold in all the information that we've learned. So I'll just say this now. I haven't finished writing the third exam. However, the third exam, I think you're going to find may in some ways seem cumulative. It's not intended to be cumulative, but basically all the content that we've talked about so far is related to what we're doing now. So for instance, I might ask you what you might do to identify whether you had a fungus, a bacteria, or a virus as a thing causing an illness. And you would need to give me some steps of how you might identify that. I'm thinking that there will be little time limit, but it'll be talking about how you would problem solve through questions related to disease. So I'll have more about the third test later on. But just as you're going through these units, I want you to be thinking about how this information connects to what we've been doing. So don't lose that connection. Okay, so let's get started with this material. So we know that there are various symbiotic relationships between microorganisms and their hosts. And a lot of times we would call this symbiotic relationships. Now, I want to point out here that we're using symbiosis in a way that may be different than how you've used it in the past. Symbiosis just means that you live together. There are various types of symbiotic relationships. Most of the time when we talk about symbiotic relationships, I think we're referring to mutualism, where both members of this relationship are benefiting. However, parasitism itself is a type of symbiosis, and in that case, one organism is benefiting, the parasite is benefiting while the host is harmed. We have amensalism, where one member is harmed, but the second member isn't helped or harmed. They're just there. And then we have commensalism, which is kind of, in some ways, the opposite of amenalism, where one member benefits while the other is unaffected. So just think about these as we talk about how microbes and other organisms may interact with one another. So we've referred multiple times over this semester to the microbiota, which your book at times may call the normal indigenous microbiota. And these are the things that live in and on our body normally. They don't cause disease. So we have our resident microbiota, which they're pretty much there all of our life. And so we would say that they have a commensal relationship with humans. But then we also have transient microbiota. So if we took a full sample of our microbiota at any point in time, and then repeated that weeks or years later, some organisms would still be there. Those would be our resident microbiota, and some would be different. And the ones that are different, we would consider transient microbiota. But then we have this group that we call opportunistic pathogens. And the opportunistic pathogens are the ones that will cause disease. Now, they can cause disease in a variety of times. And the times that they can cause disease are certainly when the immune system is oppressed or suppressed. For some reason, the immune system is not working at full capacity. There can be a variety of reasons why that might happen. It can also happen when competition is affecting certain changes, is affected by certain changes in the body. So if you remember our C. diff case, we had a disease that was being established, and then we saw the normal antagonism of taking the antibiotic by Danielle that eliminated the normal antagonism of the microbiota that should have been in her gut that would have suppressed the growth of C. diff. So that would be that situation. I think when we did that case study, you all also noted that her immune system might be suppressed because she just had surgery. And then we can also see opportunistic pathogens have the upper hand when members of the normal microbiota is introduced into an area of the body that is unusual for that microbe. So we have different microbes that live in our mouth than our eye. None of our body is really sterile. So if you had that, I'm sorry, that's kind of a gross example, but if you had saliva introduced into one's eye, you could see a normal microbe that lives in your body cause disease when moved to a new part of the body. So, this is a term that you may have heard recently because there has been a lot of conversation. At least last month, I was hearing a lot of conversation about reservoirs of infection, specifically as it relates to SARS-CoV-2, which is the virus that's causing COVID-19. So a reservoir of infection could be a living or a non-living source of an infectious disease. Now, we might consider carriers of a disease a reservoir, and usually we wouldn't consider like if someone had sneezed or coughed on a wall and droplets with a virus were there, we wouldn't consider that a reservoir of infection because it wouldn't be continuous. But we do have animal reservoirs. So there's been a fair amount of discussion about if there is an animal reservoir of this virus. And we know that animal reservoirs are present. We think about certain types of influenzas. So we would call those zoonotic diseases, ones that can move between animals and humans. So obviously that could be a direct contact. It could also be if you handled animal waste. Let's say that you were working in a lab that had mice that had been infected with a particular microbe, and as you were cleaning their cages, you could be exposed. But also arthropods. So if we think about insects like mosquitoes that can definitely carry disease as they perhaps bite an animal and then bite a human, and there can be transmission in that way. Now, we can also have human carriers that would be considered reservoirs of infection. You may have heard about this frequently about people who can carry strep. They don't get sick with strep, but the people around them tend to fall ill with strep throat much more frequently than the general population. And then we can have non-living reservoirs. And usually these would be environmental situations like soil, water, and even sometimes food. Okay, so the next group of terms are extremely important. So I'm just going to highlight them for you here. So we're exposed to microbes all the time. We've talked about how we live in symbiotic relationship with a number of microbes. Some of them live on us all the time. But many of them don't cause diseases. Okay, so let's talk about the appropriate terms we would use about exposure to microorganisms. So the first one is contamination. We are contaminated. Contamination just means that there's a presence of microbes in or on our body or a particular object. Now, we've talked about when we have sterile pipettes or sterile items in the lab, opening those means that now they can be contaminated. We have to consider them contaminated, meaning that there may be microbes on them. Now, let's contrast that with the idea of an infection. So we're contaminated all the time, but we're not always infected. So when we say that we are infected, we're saying that the organism has evaded the body's defenses, and we're going to talk about the different lines of defense, that organism is multiplying and it is established in the body. Now, just because you're infected does not mean you have a disease, okay? So you can be infected and have no symptoms whatsoever and everything is fine. So when we have an infection, we do have to think about the portal of entry. And the three major pathways that pathogens use to enter our body is through the skin, through a mucous membrane, and it can even enter a body through the placenta. Now, if we bypass those three normal routes, that can happen through the parenteral route. So think about if you step on a nail outside, you've bypassed the skin because you punctured it. That can obviously happen occasionally. And so these are ways that we can establish an infection. Now, some things have to happen for us to be infective. First, the microbe has to adhere. So it must be able to attach itself to our cells so it doesn't just get swept away, either by blood or lymph or something like that. Now, those organisms that can infect must have some type of adhesion factor. So they basically use some type of an attachment protein on their surface or on their cell membrane or cell wall that allow them to adhere. Another thing that has to happen for an infection, or before we go there, sorry. If a bacteria or a virus loses its ability to make adhesion factors, so if it were to mutate and not have the ability to adhere, or if we had a particular drug that could block its ability to make its adhesion factors, then we would make that organism avirulent, which if you remember, if you put a in front of a word, that means not, so it's no longer virulent. It can no longer cause disease. Now, if we contrast that, if we have a pathogen that can adhere during an infection, it many times will form a biofilm. So basically, if it's a bacteria, it can produce basically a loose glycocalyx, which as it does that, it allows it to adhere to the surface and oftentimes allows it to hide from the host's immune system. Okay, now remember, we've established an infection. That does not necessarily mean disease. However, disease always has an infection happening prior, okay? We can have an infection without a disease, but we cannot have a disease without an infection. Another term that basically is a synonym to disease is morbidity. So often especially in journal articles, you'll see morbidity and mortality. So that's basically saying the number of disease cases and the number of deaths. So you may be seeing that term. Remember, it means disease. And so disease is a condition that is sufficiently adverse to interfere with normal functioning of the body. So if we have an opportunistic pathogen that has set up an infection, it is altering our normal body functions. Now, a disease has symptoms. So symptoms may be that you are coughing, that you don't feel well, you are tired. Those are symptoms. Signs. The doctor can see the signs. The doctor cannot see that you don't feel well necessarily, but the doctor can see that you are coughing. The doctor can see that you have a fever if they put a thermometer in your mouth. And so they can be seen or measured by an outside observer. Those are the signs. So you'll see often people will talk about the signs and symptoms of diseases. One is only experienced by the patient. The other can be observed from the outside. So when we put together our symptoms and signs in a group, we call that a syndrome. So you will see references to influenza-like diseases. And so usually that's referring to a syndrome, which is a group of diseases that are like the flu. So fever, body aches, coughing, sneezing, runny nose, those things all together tend to group together when you have the flu, and we would consider that a syndrome. Now, we can also have disease and be either asymptomatic or subclinical. So basically, this is you're infected, but there's an absence of symptoms. Now, if we were to go through specific clinical tests like blood tests that look at white blood cell numbers, or like if we're talking about strep, if you had a throat culture done, there may be signs of infection, but you don't have the symptoms that you would expect if you had that disease. So there's actually some discussion right now about COVID-19, about the number of asymptomatic people who maybe were infected or had a subclinical infection. So they were infected, but now when serological tests are happening, they are presenting with antibodies, but they never knew that they had encountered that disease. So that is certainly another outcome that we see happen pretty frequently, that we may be asymptomatic or have a subclinical infection. Okay, so let's talk about a word you may have heard on the news, etiology. So etiology is a fancy word for what causes a disease. And so there are etiologists, and their life's work is to understand the cause of diseases. Now, this can be tied back to the history that we learned very early in our class about Cook's postulates. So Cook's postulates really led to the germ theory of disease. And remember, before Cook, we didn't understand that bacteria actually caused diseases. Because there was no way to systematically test that. We take that for granted now. We expect that if you have particular signs or symptoms, that there is something causing it, and that we expect it to be some type of microorganism. And so if you remember, we had several conditions that Cook's postulates talked about that were essential to prove the cause of a particular infectious disease. However, as we've learned more and more about microorganisms and about diseases, Cook's postulates are not perfect, and they actually have some problems. So first of all, some pathogens, we cannot culture them in the laboratory. We have tried to grow them on every kind of agar or media and every kind of condition in an incubator, and we cannot get them to grow. Remember that Cook's postulate said you needed to be able to isolate a pure culture in the lab and then infect something else. But if you can't culture it in the laboratory, that is pretty much impossible. Now, it's also true that some diseases are caused not by a pure culture or one organism, but are caused by a combination of pathogens as well as cofactors. Cofactors meaning specific, it could be specific life stages or other things that are specific to a group of people. Obviously, Cook's postulates have some ethical issues, because if we're talking about a disease that only affects humans, should we really infect humans to make sure that this is the actual causative agent? And we have seen that at times some pathogens are ignored as a potential cause of disease. Sometimes that's due to prevailing knowledge. That's what we've always known. A good example of this is Helicobacter pylori. For years, it was believed that stomach ulcers were caused by excess acid within the stomach of those patients. And it was treated using antacids. However, stomach ulcers were then found, the doctor won a Nobel Prize for this, and we'll actually, I think we're doing this case study later on about how he discovered Helicobacter pylori. But it's a very small bacteria, And that very small bacteria had evaded, because people weren't looking for a bacteria, but it had basically invaded the stomach lining, and it actually caused the ulcer. And then the stomach acid just exacerbated the pain of the stomach ulcer. So we certainly see it's possible that there are diseases or situations that we have not paid attention to those potential causes of disease, and we'll find some new pathogens. Okay, so here's more terms. I feel like chapter 14 is very term heavy, but they're important terms. So we have this idea of pathogenicity. So can a microorganism cause a disease? If it can't cause a disease, we do not call it a pathogen. However, not all diseases are created equal, or not all pathogens are created equal. So that's where we refer to virulence. And it is a measure of an organism's pathogenicity. So I think often of Ebola or other organisms that are in the BSL biosafety level 4 and 5, or even 3. They will cause disease even if a healthy person is exposed. But then we have other organisms that may be biosafety level 2. They're less virulent. A healthy individual might come in contact with them and not have any symptoms, or their immune system is able to fight it off. Okay, so why do we have this difference? I just told you, some organisms are very virulent. They can cause disease in just about everyone they encounter. Other microorganisms are not. So why? Okay, so the first thing that can happen is how effective is that microorganism's adhesion factors, and how well do they form biofilms? If they're able to adhere very tightly and easily, if they're able to form biofilms, they are likely a more virulent microorganism. We actually see this in strep. You'll hear them talk about you have a more virulent strain. There's still streptococcus, but some strains have one reason that they're more virulent is due to adhesion factors and or their ability to form biofilms effectively. Another thing that increases the virulence is the ability to produce particular extracellular enzymes. So basically, these pathogens secrete them into their environment, which if you're the one infected, they're secreting this into you, and it actually can damage and break down your cells or your connective tissue, which allows it an extra nutrient source in most cases. Another virulence factor, things that make microorganisms more pathogenic, more virulent, the ability to produce toxins. So these are different than extracellular enzymes because enzymes are breaking something down. Toxins are chemicals that are released that harm your tissue or trigger an immune response to cause damage. So we know that if toxins get put into your bloodstream, that you actually are diagnosed with toxemia. So it's actually moving. So let's say that you step on a nail, you have an infection in your foot. Toxemia can occur if that organism has released toxins and now they've made it into your bloodstream and are being carried throughout your body. Now we have exotoxins and endotoxins. So exotoxins are those that are secreted by pathogens into the environment. But if you remember, we talked about lipid A, which is produced or is present, lipopolysaccharide is present in gram-negative cell walls. And when lipopolysaccharide is broken down, lipid A is released. Lipid A is an endotoxin. And if it forms a toxemia, it actually can be fatal. It's very dangerous. So you actually see, if you have an infection of gram-negative bacteria, that you have to be very careful about a dosing of antibiotic, because yeah, we can kill that organism with an antibiotic very quickly, but if there is a high number of bacteria in the system that we kill when they're dying and dead cell walls start to shed off, lipid A is released into the bloodstream, and you can actually greatly harm or perhaps even kill your patient by the release of these endotoxins. And we also have another virulence factor are antiphagocytic factors. We have several immune cells that they protect us by eating or phagocytizing those invading cells. And so if the pathogen has different factors that basically hold at bay or prevent our immune cells or the host's immune cells from doing their job, you can see that that would increase the ability of an organism to cause a disease. Okay, here are some more terms. So after you've been infected and now you have a disease, what are the stages that we go through? So the first stage is the incubation period. It's the time between when you're infected and when you have first symptoms. So you all have been living through questions about incubation periods because one of the reasons that we are under quarantine or whatever level of staying at home and sheltering in place you would like to call it, the reason we're doing that is because you can be exposed to this virus and it can be up to 14 days before you show any symptoms. We would call that the incubation period. Then we enter what we call the prodromal period. It is a shorter time, and that's when you start to feel bad. But you don't feel terribly sick yet, you just don't feel great before we enter this time of what we would call illness. So that's the most severe stage, and your signs and symptoms of whatever disease you are experiencing are most evident. We next enter decline, where you start to feel better. Now, obviously, this is happening either because your immune system has kicked in or there have been some type of treatments available to you that has helped to kill off the pathogens in your body. And so your body is trying to return to normal. And convalescence, the pathogens are gone, but the damage that they did in terms of extracellular enzymes, toxins, tissue damage from those kinds of things, they're needing to be repaired. So your body is not completely back to normal, but you are no longer infected. So pretty much every disease follows this kind of pathway. Obviously, the timing for each of these periods varies. So you might have a very short incubation period for a particular organism. You might have a very long illness time. You might not really even feel like you ever experienced the prodromal period, and you may feel like you recovered very quickly and you weren't able to really detect the idea of decline and convalescence. But that all depends on the particular disease and the particular pathogen that you're dealing with. Now, we've already talked about the portals of, we talked about portals of entry, but we also have portals of exit. Because if we have a contagious disease, we want to know how it moves from person A to person B. So portal of entry is how it infects you, but then portal of exit is how you may infect others. So nose and mouth are two of the most common, as well as urine through the urethra. So we can have different ways that we can transmit diseases. We can have contact transmission. So you might have direct or indirect contact. I think those are pretty self-explanatory. But we can also have droplet transmission. So pathogens may travel up to 1 meter maybe in a droplet of mucus when someone is coughing or sneezing. And this is why masks are being mentioned for COVID-19 to minimize droplet transmission, because droplet transmission has been, or is expected and has been shown in the data so far to be one of the more common ways that COVID-19 is being transmitted from person to person. We also have vehicle transmission. So I don't know if any of you have ever watched the movie Outbreak. It is from the 90s, Rene Russo and Dennis Hoffman. It was actually about Ebola before all the Ebola outbreaks of recent years have brought that back to light. In that movie, the Ebola mutated to be airborne. So therefore, it was traveling long distances of people, in the same hospital. through the HVAC unit. So it's actually on Netflix right now. If you're interested, go watch it. Pretty interesting movie. We can also have vehicle transmission that's waterborne. This is why you probably have been taught to not drink water directly from streams when you're hiking. We also, foodborne can be a vehicle that transmits organisms, and then we can also have bodily fluid transmission. So this is why we use PPE when we utilize any body fluids in the lab, or why healthcare workers should have access to PPE while they're working in a healthcare setting. We also have vector transmissions. And so we've also talked about vectors in our last unit. Now, mechanical vectors are things, they're not hosts. So this can be, please understand, mechanical vector transmission might be that a fly landed on a cow that had a particular disease, and then, and actually this is, so pink eye is prevalent in cattle in the summer, and so flies like to congregate on cows. And so if one cow has pink eye, a fly might land on their eye, and then they pick up those organisms on their feet, and when they go to another cow and land on their eye, they've now transmitted the disease to the other animal. That's not a biological, where we're talking about a bite, where we have this organism that's a host and a vector. That's mechanical. It's on the feet. The fly was not really, the fly was contaminated. The fly was not infected. So mechanical vector transmission happens when you have a contamination, not an infection of a particular, of an intermediate animal. Biological vector transmission are what you think of with mosquitoes biting and flying from one individual to another and spreading disease. Okay, now more terms. Diseases can be, or infectious diseases can be grouped by the timing as well as their severity. So if you have an acute disease, it develops fast and lasts a short amount of time. Contrast that with chronic, where you have a very slow development and it's continual or recurrent. Now, a subacute disease is in between acute and chronic. The durations and severities are in between those. And then we can have a latent infectious disease where a pathogen can remain inactive for a long period of time, and then maybe a weakened immune system or something like that, it can now cause disease. So usually that is happening. That's not the same as just being a carrier, but at times it can be. So you got acute, subacute, and chronic, as well as latent infections. Now, a different grouping or way to classify infectious diseases is how it's transmitted. So we can have communicable diseases where you have one host coming directly or indirectly from another disease. Contrast that with a contagious disease. So all contagious diseases are communicable, but not all communicable diseases are contagious, okay? So a contagious disease is when you can easily transmit this from a reservoir to a patient. A non-communicable disease, they either arise from outside of the host or from the normal microbiota. So you don't have to worry about catching a non-communicable disease from someone else. I'm sorry. Okay, so you've also heard the term epidemiology a lot lately because epidemiologists are talking a lot about our current pandemic. So epidemiology is basically what those people do. They are studying where and when diseases occur and how they're transmitted in the population. So I hope that you can see why epidemiologists are very important to our current situation. So we are in the midst of a pandemic at the time of this recording. We have an epidemic of a virus that is on pretty much every continent of our planet right now. And epidemiologists are tracking the number of new cases. They're tracking the number of total cases. And from that data, that indicates what type of outbreak we're experiencing. So when you hear someone say that this is an endemic, we would say that this disease is usually present, maybe not in high numbers. So sporadic means that we have this happening occasionally, and then we have an epidemic when we have a higher than normal number of cases. So we are definitely experiencing an epidemic for this particular virus right now, but it is considered a pandemic because it is being experienced worldwide. So you have different subspecialties of epidemiology. I've included this here because it's kind of relevant to current events. So you can see that epidemiologists even can subspecialize in different areas. Okay, one important term that I would like to make sure that you know, and we've also talked about this a little bit in our C. diff case like a couple of months ago now, is the idea of a nosocomial infection or a nosocomial diseases. These are healthcare associated infections. Staph is often considered at this point a nosocomial infection because some health facilities have an increased prevalence of staph infections that move from patient to patient. We learned in our case study that C. diff is another one of these. So you can see there that there are three types of nosocomial infections present: exogenous, endogenous, and iatrogenic. And as we talked about in our case study, hand washing and other aseptic and disinfecting techniques can protect individuals from exposure to both nosocomial infections, but really any type of pathogen. The WHO uses the different epidemiological data to give rules and standards for clean, potable water, safe food, and to help prevent disease by controlling vectors in animal reservoirs. So the World Health Organization is the largest organization in the world that is concerned with epidemiology. That is their line of business. That is what they do. And they are trying to help governments around the world to be prepared to deal with any type of infection that may be emerging in our world. Okay, so let's talk about some of the ways that we do not get sick. So we're going to kind of go quickly through these slides. I will stop at the end of each chunk and confirm if you do or don't have questions and answer those specifically. If you've had A&P, some of this may be very familiar to you. If you've not had A&P, I hope some of it's still familiar to you. Maybe you've been introduced to it previously. So overall, when we think about how we do not get sick, we have two ways that doesn't happen. First, we have our innate immunity, which that is what we're born with. There are certain things that do not make us sick or cannot make us sick for two main reasons. And that is either we have external physical barriers that don't allow the pathogens to set up an infection, but then we also have immune cells and chemicals within our body that if a particular organism gets past those physical barriers, like our skin, it is able to kill them and rid our body of them without a problem. But then we also have adaptive immunity, which we consider our third line of defense. And so this response is specifically generated against each particular antigen. So adaptive immunity is the reason why we say that if you get sick with some particular diseases, think of chickenpox, for instance, that you will not get chickenpox again in most cases. So we're going to talk about how that works. Okay, so the first thing here, we have the skin, and I'm not going to read through all of these areas of the skin. Unbroken skin is one of the best, it's one of the best barriers against disease, and you can see the different components of the skin, and each of these components helps to either inhibit the growth of bacteria or prevent the adhesion of bacteria so it cannot enter our skin. There was one semester that I actually had burned my hand in the spring semester. And so we were in microbiology lab, and unfortunately I had to wear a glove all semester because there was no way for me to protect my hand in microbiology lab. And I had an open wound, so my skin was compromised. I couldn't just wash my hands. But that's why we can wash our hands in our lab and not have to worry so much about disease. Now, with also another component of our first line of defense are our mucous membranes. So you might think about the membrane inside of your mouth. And so there are multiple layers, and similar to the skin, they are acting to trap and remove pathogens before they can set up a disease. And this extends to your eyes, your saliva, and then we also have this idea of microbial antagonism, which you've already been introduced to in the C. diff case, where we have competition between the normal microbiota and potential pathogens present in any area of our body. Okay, so basically those are mechanical barriers. Those mechanical barriers can include both structures of your body as well as secretions of particular structures of your body, thinking of saliva and tears. So if you get past the first line of defense, so you've penetrated the skin or the mucous membrane, we now have a second line of defense. So we have several immune cells that are white blood cells that are primarily phagocytes. They are scavenging your body right now looking for invaders. We have different antimicrobial chemicals. I've got a list of them here. I'll mention like lysozyme, that's within both our tears and our saliva, that are antimicrobial in nature. And then we have these different processes that help us to fight off disease. So we need to talk quickly about blood as a part of the second line of defense. So the liquid part of the blood is plasma or serum, depending on whether the clotting factors are present. But then we have the formed elements. So red blood cells are carrying oxygen and carbon dioxide. They are not involved in these processes. But then we have this large group of white blood cells or leukocytes. And they are grouped into two different types. We have the granulocytes and the agranulocytes. So basically, we are dividing them based on how they look under a microscope. Granulocytes have large granules that stain different colors under the microscope. Agranulocytes don't look that way. So the granulocyte groups are basophils, eosinophils, and neutrophils, and I've given you how they stain here. They each have a different role. And then we, for agranulocytes, have lymphocytes and monocytes. So basophils release histamine during an inflammation, which actually calls more immune cells to it. Eosinophils and neutrophils actually will phagocytize pathogens. And they can all actually move in and out of our capillaries by diapedesis, which is a process where they actually squeeze between the tight junctions in the cells out into another part of the body. There are some really cool YouTube videos that are showing either simulations or that this actually happening. Feel free to look those up. And then we have macrophages, neutrophils, and the dendritic cells, which are also phagocytic cells. Now, you may be thinking, oh, we'll get there in just a second, sorry. Included in this, we can do, basically to see if you're sick, we can analyze the types and numbers of leukocytes or white blood cells. So we can actually do a differential white blood cell count that will tell us how many white blood cells and which types of white blood cells. And that actually gives us some clues as to what disease you may be suffering from. And actually not even a disease, what pathogen you may be encountering. So we know that we have chemokines and other chemotactic factors that are attracting our phagocytic leukocytes to the site of damage or invasion. We know that we can have opsonization, which basically is a way that some types of cells in our body can mark invaders for phagocytosis by other leukocytes. And so what's really cool though is these white blood cells I'm talking about, they're going around and they're phagocytizing cells. How do they not phagocytize their own body? So the reason we consider this the second line of defense is these particular leukocytes are able to recognize the surface proteins of cells. And so they know, here are what our surface proteins are. And what they are able to then do is we have a number, tens of thousands, I want to say it's maybe 20,000 genes that have been identified. That might be too high. But we'll go with it for now. We have genes that are normally within our genome that are coding for what we would consider invaders and their surface proteins. So these white blood cells are able to identify invaders based on genetic signals that we have the ability to make antibodies against almost immediately. And antibodies can be involved in this process of opsonization and also in the ability to recognize an invader and then send out these chemokines and chemotactic factors. Now we also have some of these white blood cells that actually don't kill anything, but they have a role in our second line of defense. So you can see eosinophils can be involved in cases of both cancer as well as helminth infections. We have natural killer lymphocytes that actually secrete toxins. So they don't eat them, they secrete toxins onto the surface of our cells that might be infected by a virus. And actually, those of you who have heard of Dr. TG, her dissertation work actually looked at natural killer cells and how that might actually be an appropriate cancer treatment. And then we also have neutrophils that can use the toxic forms of oxygen, and they can also form what we call nets, neutrophil extracellular traps, which actually do kind of look like a net if you look at them under the microscope. So they trap and contain bacteria or other microorganisms rather than actually phagocytize them. We also have the PAMPs. So pathogen-associated molecular patterns, which are connected to our toll-like receptors on the host cell membranes and nod proteins inside of cells. I'm going over these because I want you to understand that they're the second line of defense, but much like in our biochemistry unit, I'm not going to ask you to draw out all the steps. I do want you to understand which of these steps or which of these possibilities fit into the first, second, or third line of defense. Okay, we also have interferons. I've given you some details about the different types of interferons that are involved in our second line of defense. And then we have this idea of the complement system. And so I've mentioned here some words that we've seen before. They can trigger inflammation and fever. They can act as a chemotactic factor that can bring other phagocytic cells in and can destroy foreign cells with these MACs, the membrane attack complexes, which basically can rupture invading cells. And then we have multiple ways that we can activate that complement pathway. Again, this is part of the second line of defense. These pathways can all occur the first time we encounter an invader. Our body is able to recognize them immediately and wipe them out. Okay, we've talked a little bit about inflammation and fever. You all know what these are. But inflammation actually occurs. And one good thing about inflammation is we have increased blood flow to the site. So one, it obviously causes us hopefully to not move that a lot because we don't want necessarily movement out of the area because we want the invaders to stay in the area of inflammation. But we do want excess blood flow to go there so more of our leukocytes are present to fight them off. Fever is happening because there have been pyrogens released a lot of times by our cells, but they can also be released by the bacteria. One thing that's really interesting is that fever is a protective mechanism because most bacteria, or a lot of bacteria, maybe not those that grow in the soil, but a lot of bacteria like to grow at 37 degrees Celsius. Far fewer of them like growing at a higher temperature. So fever is actually one way, even though the pyrogens may be released by the invader themselves, but it's one way that we actually decrease the growth rate of the bacteria because now we have exceeded their optimal temperature for growth. Okay, now, like I said, everything in the first two lines of defense is you're born with. It's there because you're a human. It's coded in your genes. You can recognize those invaders that we can stop with the first two lines of defense at the first time we encounter them and every time thereafter. However, we also have this thing called adaptive immunity. And adaptive immunity is a lot more specific, but we are not born with this immunity. So adaptive immunity allows us to recognize and defend itself against new invaders, distinct invaders, ones that we do not have the normal genetic capability to respond to, which when you think about one, how many possible pathogens there are in the world, as well as how those pathogens themselves can change and mutate, it is good that our body has a way to respond to that. So adaptive immunity is even more specific than the second line of defense that we were talking about. And adaptive immunity can be turned on and off, so it's inducible. It can ramp up production, so it has this clonality. It is unresponsive to our own self, which is also a good thing. We do not want our own immune system to attack us. And adaptive immunity has memory. So once you've been exposed, you have the ability to respond immediately and not be in a disease state again. So the main leukocytes that are involved in adaptive immunity are the lymphocytes, the B lymphocytes and the T lymphocytes. Now, the B and the T are coming from where these cells matured. So you'll see B lymphocytes mature in bone marrow and T lymphocytes mature in the thymus. They have different roles, although some of those do overlap. So let's talk about adaptive immunity. So this is our third line of defense, adaptive immunity. So one part of our adaptive immune system is the lymphatic system, which if you've taken A&P, you should know that we have a system of vessels that pick up lymph, which is basically the same as our blood plasma. And we also have lymph nodes. So this is a one-way system that goes from basically our tissues back to the vena cava right before blood enters the heart. So we consider not just the lymph nodes in the vessels, but also our thymus, spleen, tonsils, and then what we consider the MALT system as part of this lymphatic system. And basically the lymphatic system is screening the tissue for foreign antigens, things that we just don't recognize and we don't think are our own self. So how this happens are we have antigens. Everything has antigens on the surface. So if we looked at viruses, microbes, dust, dirt, under the microscope, it has a particular biochemical composition, and therefore that the biochemistry, the atomic structure on the surface is considered its antigen. Now, in this case, we are mainly talking about antigens that will actually trigger a specific immune response. So pollen, for a lot of people, has a surface protein, glycoprotein, or molecule that triggers an immune response. That's why you have itchy, watery eyes. That's why you sneeze, and can have some pretty severe reactions to pollen. We consider those antigens. So basically an antigen, our body says this isn't us and we need to attack it. Now pollen is not going to do anything to us. Pollen is not alive in the sense that it can multiply in our body. But our cells clearly don't know that, so they are attacking it. Now, if you have an effective antigen, they're large, complex, stable, degradable, and they're foreign to the host. So therefore, you don't have cross attack where the immune system has recognized something that is actually you and attacks it like it's foreign. So effective antigens need to be recognized as foreign. So we also have this piece of the antigen called the epitope. That's the three-dimensional shape of the region of the antigen that is recognized by the immune system. So the whole antigen isn't recognized. It's just a specific piece of it, and we call that piece the epitope. Now, we can have two different types of antigens. Exogenous antigens are found on microorganisms that multiply outside the cells of the body. So maybe they are multiplying within the gut. We also have endogenous antigens. Those are inside of our body cells. And then we actually can have autoantigens. Basically, our cells are covered by different glycoproteins and proteins. And so basically we have this process of self-tolerance. So our body does not attack the autoantigens. Now, if you read in chapter 17, it talks about when that actually goes wrong. And when this process of recognition of autoantigens goes wrong is when we see people develop autoimmune diseases. But that's in chapter 17, and we didn't have time to cover that in our semester. Okay, so we've already talked about that our adaptive immunity is mainly composed of B cells and T cells. So let's talk about T cells. Again, they're made in the thymus. They act primarily against the intracellular pathogens. They're constantly circulating in our lymph and our blood. And they have T cell receptors on their cytoplasmic membranes that have an antigen binding site for a very specific epitope. So T cells can only, so let's say that you've been exposed to chickenpox and there are specific T cell, chickenpox T cells, meaning that they can recognize the chickenpox virus and have a specific antigen binding site for that specific epitope. T cells are split into two different groups. We have cytotoxic T cells, which I've given you the way these basically you'll see the book refer to them both of these ways. Sometimes they'll call them TC cells. Other times they'll call them CD8 cells. I'm going to call them cytotoxic T cells, but just know that these are the same thing. They can act against infected or abnormal body cells, which might include virus-infected cells, bacteria-infected cells, some cancer cells. And then we also have helper T cells that can direct either cell-mediated immune responses. A cell-mediated immune response either is asking for help from the second line of defense and the phagocytic cells, or it can direct a specific antibody response. And again, they will sometimes in the book be called TH cells. Sometimes you'll see CD4 cells as well. Okay, so we have this ability to do clonal deletion. So basically, if some of the cells that are actually going around and binding to epitopes, and then they kind of present those antigens and say, okay, do we need to make more lymphocytes to fight against this particular antigen? Sometimes it can make mistakes. And so we actually have the ability to delete the ones that maybe have mistakenly found an epitope of our own. You'll see that when that happens, that those cells are programmed to then die. So they recognize that they fall on their sword. They have realized that they are harmful to their own self, and they undergo apoptosis. So this process of clonal deletion is another way that even though our immune system is very active, that we don't attack our own self. Okay, so B cells, as we already talked about, they mature in the red bone marrow. They're mainly found in spleen, lymph nodes, and the Malt system, and they make immunoglobulins. So an immunoglobulin is this Y-shaped molecule that has two light chains, two heavy chains. I have a picture of one in a minute that we'll look at. And so we can have B cell receptors that actually insert a immunoglobulin into the cytoplasmic membrane, and immunoglobulins are the same thing as antibodies, everyone. So antibodies and immunoglobulins are basically free immunoglobulins. So we have B cell receptors where we have an immunoglobulin in the cytoplasmic membrane of the B cell, and it's held there by the transmembrane proteins. But then we actually have antibodies that are secreted onto the cytoplasmic membrane of B cells. So let's actually talk about what those look like here. So this is an antibody or an immunoglobulin. So this is basically a showing you the actual protein structure with the beta pleated sheets and the alpha helices that might be present. And the disulfide bonds are kind of evident in here, but we have the two light chains here and the two heavy chains, all right? Now, up here are where we bind to antigens, all right? So when we look at the pink and the red down here at the bottom, these are the same. But these ends, the light chain, up in the end of the light chain, the end of the heavy chain, these are variable. They're different per antibody or immunoglobulin. And that is why they can be specific to specific antigens. Okay, so that variable region on the end is actually the antigen binding site. And that's what allows it to recognize and bind to those regions, the epitope regions of invaders or pathogens. Now, there are 5 classes of antibodies. I want to say there's a picture in your book that shows you the difference between them. You may have heard of these before. So we have IgA, IgD, IgE, IgG, and IgM. Now, this is based on differences between their stems and the type of heavy chain that they have. I'm not going to give you pictures of an antibody and ask you which of the 5 types is this, okay? I just want you to be aware that the 5 types exist and have some basic information. I mentioned earlier about allergies. So when you have spring allergies to pollen, IgE is what is happening. Or if you have another type of allergy, IgE is what is triggering that allergic reaction. Okay, so sometimes we have these cell-mediated immune responses. So I've given you some different details. I've summarized some of the details here. But basically, what is happening in any of these cell-mediated immune responses is the cells themselves are coming in and are having an action. So it might be that the cell has extracellular enzymes where like perforins and granzymes that can kill the invading cells. It may be that they can recognize abnormal molecules. It might be that the cell-mediated immune response calls additional lymphocytes in and actually forms both memory cells and makes a lot more of the TC cells, the cytotoxic T cells, so it can go and kill more of the invader. So obviously you see cells themselves coming in and killing and containing the pathogen. Now, I want to back up just one second. When we have our antibody responses, those are not directly related to cells, although the antibodies are made by white blood cells. But in those cases, The antibodies are made by the cells, and then the antibodies are the ones that are using particular processes to contain or kill the invader. So please keep in mind that you've got antibody-mediated responses, which use the antibodies as we talked about, versus the cell-mediated responses. Now, there's been a fair amount of conversation about the topics on this slide, and so I wanted to highlight them. We have multiple types of immunity, okay? So when I, as a child, had chickenpox, and I did have chickenpox when I was 4, I developed naturally acquired active immunity. So I got sick because I was exposed to someone who had chickenpox. So that is a natural acquisition. My body, my immune system, then had to fight off the chickenpox virus. So that is an active process. So I have naturally acquired active immunity against chickenpox. We also have naturally acquired passive immunity. I worked on a research project when I was in college that looked at, so dairy calves are taken away from their mother very early. They are fed colostrum, which contains a lot of IgG and IgA in the milk to the baby. However, those calves tend to get sick more frequently. And so basically the cow's stomach, for several days in mammals after birth, IgA is a big molecule. And for several days after birth, IgA can actually be absorbed across the digestive system into the bloodstream. So they were actually looking at ways to protect IgA in the colostrum that they were feeding baby calves in hopes of boosting their immune system. Now, that is naturally acquired because they were eating it. However, it's passive because you're being given antibodies that your body did not make. So that's a naturally acquired passive immunity. Now, because of the great strides we made in medicine, we now have the ability to have artificially acquired immunities. So when you go get shots or vaccines, that is an artificially acquired immunity. And in most cases, that is an active immunity. Usually you're being injected with a weakened form of a particular pathogen, or maybe not even the whole pathogen, just the outer coat, the antigen piece, the epitope of that particular microorganism or disease. And in any case, we're injecting you with something that causes an immune reaction. Your immune system then believes it's being invaded by something that can hurt it. And then it begins to make those memory cells for cell-mediated processes. It also begins to make antibodies that are also stored in other memory cells, so that when you may be exposed to actual invader, like hepatitis, when you're exposed to hepatitis in real life, now you can respond to hepatitis immediately rather than getting sick with hepatitis and only being kind of like me with chickenpox. You all actually have artificially acquired active immunity against chickenpox. You didn't have to go through the disease. So if you encounter the chickenpox virus, we all now can respond. But I do it because I have a natural acquisition. You do it because our bodies went through the same process, but your immunity was artificially acquired. Now, we also can have some artificially acquired passive immunity. So we can either inject you directly with antibodies, kind of like similar to a baby calf drinking its mother's colostrum, which is rich in IgA. We can give people antibodies in antitoxins or antisera. And actually, that is one line of study right now with COVID-19, because they've been asking for people who have been infected that are showing antibodies, would they be willing to donate blood so that they could actually dose individuals with the plasma or serum of someone who's already been infected, those antibodies would be present in the plasma or serum and therefore would help that patient to fight off COVID-19. This has happened much less. Most of the immunizations that you have had so far in your life would be artificially acquired active immunity rather than passive immunity. However, since we don't have a vaccine against COVID-19 or this particular strain of coronavirus, we're trying this passive immunity option, which actually, from what I understand, is what often happens when you have an emerging disease. We try options for artificially acquired passive immunity while we're working on some type of vaccine to allow for artificially acquired active immunity. All right, I didn't stop in between because I forgot. So I'm going to stop sharing my screen. Do you all have any questions? I feel like I just fed you with a fire hose and we went through a whole lot of information. But I hope it was a quick summary. Please remember, I will not ask you to draw out any of these processes. The most detail I'm going to ask you to go into is to outline the first, second, and third line of defense. You need to know what's involved in each one of those lines. You need to be able to list out some of the components of each one of those lines. And if I gave you one of those items, you need to be able to tell me it's a part of the first, second, or third line of defense. There was also, as I mentioned, a lot of vocabulary in chapter 14, and you do want to familiarize yourself with those vocabulary terms that we talked about. There were several. But do you have any other questions? Okay, if there's no questions here, I'm actually going to stop the recording.